You ready?
Yep.
Okay, go right ahead.
Okay. Hi, everybody. My name is Claire Liu, and today I'm going to be presenting on my project investigating stress response genes in C. elegans to prevent Huntington's disease. In my presentation, I'll be starting off with a little bit of background on Huntington's disease, including the specific model organism that I used and the target genes that I investigated, followed by my hypothesis and the 3 experiments I conducted to test this hypothesis, and finally ending with my results and a discussion of those results. First off, a little bit of background on Huntington's disease. I became really interested in studying neurodegenerative disorders when a really close friend of mine, family friend of mine, was diagnosed with one. Huntington's disease is a genetic neurodegenerative disorder, and it's characterized by uncontrolled movements, speech difficulty, and memory loss, among a couple other common symptoms. Now, genetically, Huntington's is caused by a CAG mutation in the HTT gene, and this causes an abnormally elongated Huntington protein, which tends to break down into smaller toxic fragments and clump together, forming aggregates. And these aggregates will lead to neuron dysfunction and eventually neuron cell death. And currently, there's no cure to Huntington's disease. But in order to work towards finding one and investigating the molecular mechanisms behind the disorder, we can use the model organism C. elegans, and specifically the EEC103 strain. C. elegans are transparent nematodes, or worms. They look like this, and they're really great to use for model organisms because there is much genetic overlap between humans and C. elegans. Now, the EEC103 strain is a specific type of C. elegans that has been genetically engineered to contain human mutant Huntington protein expressed in their body wall muscle cells. It's also tagged to YFP yellow fluorescence, as you can see in this image right here, which makes it easily visible. And lastly, we can use RNA interference, which is a technology that's able to knock down specific gene expression in C. elegans by feeding them double-stranded RNA expressing E. coli. In my project, my 2 target genes are DAV16 and FA4, and I chose these based on an initial targeted screen of C. elegans, which showed that the individual knockdown of these 2 genes caused a significant increase in Huntington protein expression, and I wanted to find out why. So a little bit of background, both of these genes encode for transcription factors, which means that their proteins go on to regulate the activation of other genes. They're also both human gene orthologs, and that means that there are very similar versions of FA4 and DAV16 in us humans. In FA4, among its many functions, it promotes longevity, specifically under its activation in starvation in worms. Now, similarly, DAV16 also promotes longevity in its activation under oxidative stress. And it's also been shown to possibly regulate FA4. So this is a schematic that I drew to display the information that I just mentioned, where dietary restriction and oxidative stress in worms leads to an upregulation of longevity genes, and much research has been shown that an increase in longevity decreases Huntington protein aggregation, though the specific mechanisms through which this happens are largely speculative. And thus, my hypothesis is that the prevention of mutant Huntington protein aggregation in C. elegans through stress response requires both FA4 and DAV16. The first experiment I did to try and test this was a starvation assay, and this was done to investigate the roles of our target genes under dietary restriction and the effect on protein aggregation. To quickly go through my procedure, I had 4 conditions of worms. L4440 is just the standard negative control regular C. elegans, and also 2 plates of individual knockdown of FA4 and DAV16, as well as a double knockdown of both of these genes. I had 2 plates per condition, and one set of these plates was subjected to an 8-hour starvation. After that, I compared the starved and the control plates to analyze their fluorescence, specifically in the head region, and that's shown through little tiny dots, which is known as punctae. These are the images that I got after the starvation assay, and after qualitatively observing them, the images with yellow text are the ones that I thought to have more fluorescence in their head region. One thing to note, I did have a little bit of trouble with the fluorescent imaging machine, so that's why you'll see a little bit of difference in exposure throughout the images, which is definitely a possible room for error. And I was able to turn these qualitative observations into quantitative results using a program called ImageJ. And ImageJ works to analyze fluorescent intensity over a given specified area. And so from this, I was able to create a bar graph of this data. Upon analyzing it, we can see that the normal response to starvation is a decrease in Huntington protein or fluorescence. And although this was also observed with the FOF4 knockdown worms, we can see an overall increase in the amount of fluorescence. Furthermore, with DAF16 and the double knockdown, we can see an abnormal response to starvation. And this suggests that both FOF4 and DAF16 are required for a normal starvation response to decrease Huntington protein aggregation. The second experiment I did was an RT-qPCR, and this was done to try and map out the possible signaling pathways that involve FOF4 and DAF16. As I mentioned earlier, DAF16 is a suspected regulator of FOF4, and I also wanted to test its effect on genes that we do know that are downstream of FOF4, which are LAG2 and SOD2. And additionally, I wanted to test if RSKS1 is involved in this pathway because it is suspected to be downstream of FOF4, and it's also a gene that's really important in regulating protein synthesis and protein aggregation. In this procedure, I had 6 samples, the same 4 conditions. However, we just doubled up on the L440 and the double knockdown due to a surplus of resources, followed by RNA extraction, synthesis into cDNA, adding our 4 primers, and then conducting the RT-qPCR. And from this test, we were able to quantify gene expression and turn them into bar graphs. In the FOF4 expression bar graph, the data suggests that DAF16 is a regulator of FOF4. In the LAG2 expression bar graph, we can see that DAF16 also appears to regulate LAG2, but possibly through FOF4. In the SOD2 expression graphs, interestingly, DAF16 appears to inhibit SOD2 expression, even though the FOF4 effect seems to be dominant when we look at the double knockdown. And lastly, with RSKS1, we also had a surprising result where individual knockdown of FOF4 and DAF16 decreased RSKS1 expression. However, the double knockdown increased its expression. And this is definitely something that I would like to investigate further. From this data, I was able to draw a possible signaling pathway involving these genes of interest. One thing that is not noted on this diagram is the piece of information that I just mentioned, where the double knockdown increased RSKS1 expression. This is something that I'd like to look more into before making solid conclusions. But one hypothesis that I have regarding the cause is that perhaps a double knockdown of FOF4 and DAF16 caused a protein imbalance, and so RSKS1 was upregulated to try and compensate for that. Lastly, in the oxidative stress assay, I investigated the roles of FOF4 and DAF16 genes in oxidative stress in worms and the effect on Huntington protein aggregation. Again, I had the same 4 sample conditions, this time 3 plates per sample. One set of plates was added, or one set of plates had M9 added to them, which is just a regular buffer that's sometimes used to store worms, so it does not induce oxidative stress. And the other 2 sets of plates had different concentrations of hydrogen peroxide added to them, 0.01% and 0.05%. And after 45 minutes, I imaged them again to analyze fluorescence. Now, unfortunately, after the 45 minutes, when I looked back at the worms, I had no surviving worms in the 0.05% hydrogen peroxide solution. And I also had varying degrees of survival rates in the 0.01% solution. And this resulted in inconsistencies in sample sizes. So this would definitely be another possible room for error. However, I still imaged the surviving worms and looked at which ones seem to have more fluorescence qualitatively. From this data, I concluded that phosphor seems to be required for a normal
2
response
minutes.
for worms under oxidative stress. And even though the DAF16 response seemed to follow the normal response, in the double knockdown, it seems that the phosphor knockdown is dominant. Lastly, I'll end with a discussion of all my results. Starting with the starvation assay, my hypothesis proved to be correct that both phosphor and DAF16 are required for Huntington protein decrease, and I'd love to do additional trials in the future to confirm these results. In my RT-qPCR, I drew up a possible signaling pathway and found that DAF16 does indeed regulate FOXP4. In the future, I'd love to do more RT-qPCRs, perhaps investigating entire WAG and SOD gene families, as well as looking into the role of RSKS1. And lastly, with the oxidative stress assay, I showed that FOXP4 is required for Huntington protein decrease. However, additional trials are absolutely necessary with a decreased concentration of hydrogen peroxide to ensure a more stable sample size and more accurate and credible results, as well as maybe looking into survival rates to see if there is any correlation between the gene knockdown and survivability. But all in all, I hope that my results will contribute to further understanding of the molecular mechanisms behind Huntington's disease and lead to perhaps finding a cure in the future. These are my works cited, and thank you so much for listening.
Thank you very much, Claire. I enjoyed your presentation.